Okie dokie. So this is week 3. We're in fall 2020, 208. We're still working our way through chapter 20, which is our heart chapter. And we need to talk about, now that we've gone through what happens in one nodal cell, what happens in one contractile cell, and the changes of the electrical system, what starts to happen when you put multiple cells together and you have them work together to then interact with many contractile cells and get them to contract. All right. And so when we look at our nodal cell populations, there are some different nodal cell populations scattered throughout the heart, and they are going to work as a unit to spread the electrical signals to all of our contractile cells and then coordinate that contraction of the ventricle happens after the atria have fired and filled the ventricles and topped them off with as much blood as possible. And then the ventricle contracts from the apex towards the superior region where the valves are located to push our blood against gravity up into the pulmonary trunk or up into our aortic arch. Okay, so the conduction system is considered to be the SA node, and your SA node is going to be the main pacemaker cells. Okay, and so when we looked at our conduction system or our nodal cell, action potentials with phase 0, phase 3, and phase 4, those were most likely the image representations of the SA nodal cells. Okay, so we know that the SA nodal cells have that pacemaker potential where they have those funny sodium channels that let sodium leak in, causing that slow depolarization towards threshold. We know that the phase 0, sorry, my 0 here, is during the action potential is rapid depolarization of calcium ions coming in. And we know that our repolarization is going to be potassium ions leaving the cell to try to compensate for all these positives came in and I need a positive ion to leave. Okay, so the SA node is going to be, again, our collection of those nodal cells that have the fastest pacemaker potential to threshold. So they are generally the area of the heart that is going to set the rate at which the heart beats. Okay, and they are going to be connected to some other, again, nodal type cells that are forming these internodal pathways that are going to run the space into the right and left atria to get the right and left atrial contractile cells to contract in unison so they can, during atrial systole, force more blood by contraction and squeezing on the atrial cavity shape to become a smaller cavity. They are going to push blood into the ventricles. Okay, now the next big nodal cell population is known as the atrial ventricular node, the AV node. Now these nodal cells, if we were to look at their specific phase 4, phase 0, phase 3, their action potentials, they might look a little different from the SA node, but for our purposes this semester, we're just going to say they're about the same. Okay, now that AV node is going to receive the information from the internodal pathways, from the atrial contractile cells nearby, and because, again, cardiomyocytes are chemically and physically touching each other, that is going to help get these AV nodal cells ready to hit threshold and ready to then send the electrical wave of energy down the AV bundle, down the bundle branches, through the moderator band, and then through the Purkinje fibers to all of the cells of the right and left ventricle to get all of those ventricular contractile cells to undergo their action potential. And remember, their action potential is going to be the one where they have phase 0, phase 1, phase 2, phase 3, and their phase 4 is that flat line. Okay? So the point of the conduction system is to coordinate that the atria and the ventricle contractile cells contract in unison, but the atrial cells go first. There's a slight delay to help push all the blood into the ventricles. And then the ventricle cells start to contract. But as they start to contract, the papillary muscles pull close the AV valves by pulling on the chordinae tendineae. So the blood that's in the ventricle will not be able to go back into the atria. The ventricle cells will start to depolarize and contract from the apex first and then spread up and around. So when I'm contracting my ventricle is getting smaller in size, but that's because the apex, the bottom portion of the ventricle, is being pulled up towards the AV valves and the semilunar valves. Question, the AV bundle the same as the bundle of His? Yes, they just come under different names. Remember, sometimes names that are based upon someone named it after their dog or themselves. The books are trying to move away from those names like the mitral valve, and they're trying to name it more specific to anatomical location and anatomical feature. All right. So when we look at the EKG, again, the way the EKG is set up, lead 2, which is going to be reading the electrical changes from the right arm to the left leg, that is normally going to line up geometry-wise with the heart, the SA node to the apex of the heart. And so lead 2 is normally our best lead to best capture and see the changes that are occurring along the atria first, then the ventricles, and seeing the changes of one cell having sodium in and calcium in. Multiply that out, that you're seeing all of these cells do that, and you're capturing that change across the skin, across that the electrode from attachment at the right shoulder to attachment at the left leg. Okay, so that's kind of the conduction system of the heart. All right. And again, when you look at it in a nice step-by-step process, when we are in phase 4 across everything, that means the ventricles are relaxed. All of your contractile cells are doing nothing. There is no actin and myosin interactions. Troponin and tropomyosin are covering actin's binding site. Calcium is in the sarcoplasmic reticulum. The SA nodal cells and all of our other nodal cells are slowly depolarizing, but the SA nodal cells, more importantly, are going to slowly hit threshold first. And when they hit threshold first, that is going to generate an action potential. And remember, the action potential means that calcium comes in. We have a rapid influx of positive ions. The calcium inside the cell is released from the sarcoplasmic reticulum. And very quickly, this wave of depolarization is going to spread not only from the SA nodal cells, but to those internodal pathways and all of the atrial contractile cells that they are touching or chemically linked to through those intercalated discs. And so as the purple thing is showing, within a few milliseconds, the entire atrial cells, every cell in the atria, is depolarizing through phase 0. Following depolarization, because calcium is now in the cytoplasm of these cells, calcium is binding to troponin. Troponin pulls tropomyosin off of actin. Actin and myosin interact, and with ATP present, we start to see sliding filament theory happening. We are going to see very quickly the depolarization is going to be followed by contraction. And when the atrias contract and their size of their chamber decreases and they force blood into the ventricles, that is known as a period of atrial systole. Okay, now the atrias are going to go through their repolarization, return to their phase 4. That is going to be atrial diastole. That is going to be atrial repolarization. And while that is happening, the AV node is going to then hit its threshold. It's going to force the bundle of His, or the AV bundle, the bundle branches, the moderator band, the Purkinje fibers, to quickly spread that wave of depolarization down through the interventricular septum to the apex, and then up and around through the muscles of the heart walls, the ventricle walls. And those ventricular cells, again, are going to go through, again, their rapid depolarization, their plateau, and we are going to see, again, all of that calcium coming in during the plateau, causing calcium release from the sarcoplasmic reticulum, the depolarization event is followed by a contractile event. And when the ventricles contract, we call that ventricular systole. All right? And the ventricles are in systole, the atrias are in diastole. The atrias are not putting any blood into the ventricles because the AV valves are closed, and the AV valves are closed, remember, because the Purkinje fibers, the moderator band, has made sure not only the ventricular cells are contracting, but the papillary muscles are contracting, and they're pulling on those chordinae tendineae straps, keeping the AV valves from letting blood go back into the atrias. And as the chamber gets smaller in size, all right. So if you can see, if this is my soda bottle is a ventricle, if my soda bottle with this fluid up to the top label, if I make this chamber smaller, that fluid is going to get condensed into a smaller chamber. And that fluid, as you condense it into a smaller chamber, is going to then be pushed more forcefully on the walls as the chamber gets smaller. That's going to mean the pressure in there is going to increase, and when it increases enough that it's greater than the pressure in the pulmonary valve on the right side, greater than the pressure on the other side of the aortic valve, those valves, those semilunar valves will open and blood will start to eject out. Okay? I like this video, or I like this cartoon, because it shows you how in purple, when it's purple, that is when you are seeing in those cells the phase 0, the phase 1, the phase 2 of changes in membrane potential from a negative value of resting to a more positive or less negative value of depolarization is happening. And again, depolarization in these cells represents, in many cases, the calcium has come in at some point, and calcium is getting calcium release from the sarcoplasmic reticulum. And so in a few microseconds, you're going to start to see, following a depolarization of a membrane, contractions start to happen. Okay? Now, the cardiac cycle is what we call what is happening following the electrical changes. So the EKG is going to capture and show me electrically what is happening in my contractile and my nodal cells. My cardiac cycle is my physically seeing the contraction of the cells of the atria and of the cells of the ventricle. And it's seeing the state of those cells contracting or relaxing. Okay, so when cells of the heart are in a contractile event, they are in an event where they are rapidly depolarizing, undergoing action potentials, having calcium released, having actin and myosin interactions, they are in a state of systole. When contractile cells are not contracting, actin and myosin are not interacting, there is no rapid depolarization occurring, they are in a cell state of diastole. Okay, and when a heart goes through a cardiac cycle, the atrias and the ventricles are not going to be in atrial and ventricular diastole and atrial ventricular systole at always the same points in time. So to be able to communicate what is happening in a cardiac cycle, you need to be able to effectively communicate when our heart atrial cells are in diastole versus systole, and when our ventricle cells are in diastole versus systole. Okay? And so if we use our EKG and if we use the electrical changes to tell us when we're going to see physical changes in the heart, a cardiac cycle is going to start during a P wave on the EKG. A P wave, remember, represents when the SA node has initiated rapid depolarization of all of the cells of the atria, and all of the cells of the atria start to contract. So a cardiac cycle, time point 0, shown here, is when we see the atrial contraction occurring, following again atrial depolarization. So again, if this was an EKG, this is going to be in the middle of the P wave. Okay, and we see that because the ventricles are still in their phase 4, still in their resting membrane, that the ventricle cells are not contracting yet, they're not doing anything yet, they are in ventricular diastole. Okay, so the first step of a cardiac cycle is going to be atrial systole. Atrial cells are contracting. That contraction is going to push blood through the AV valves into the ventricles, topping off and filling the ventricles with a little bit of extra blood. The ventricles at time point 0 are not doing anything. They are in a relaxed or diastolic state. Now, the atria will eventually start to relax, and we will then see that when they start to relax and we no longer see active contraction of the atrial contractile cells, they are going to enter their diastolic period. Their diastolic period means that they are repolarizing, they are resetting, they are going into the end of phase 3, mostly phase 4 of their resting membrane potential cycle. Okay. The AV node is going to initiate ventricle contraction and ventricle depolarization. So we are going to see that the atrial systole ends and the ventricle systole begins. Now, the ventricle systole period is going to be divided into 2 phases. Phase 1, or A as I'm writing here, is going to be the ventricles are in systole, but we are in a state of isovolumetric contraction. And that means that the ventricles are starting to contract and the volume of blood inside the ventricle is not changing. And the reason why it's not changing is the AV valves are closed and the semilunar valve is closed. So there's no place for the fluid in the ventricles to go. So that it's iso, meaning it's not changing, volumetric, so the volume is not changing, but I have contraction of the ventricles. Is the ventricle chamber getting smaller? Yes. Are ventricle cells contracting? Yes. But as they're contracting in this small period of time, no volume is coming in and no volume is coming out. The volume of blood in that ventricle at that time is getting squished into a smaller chamber. And that is making the pressure that fluid is exerting, that push of the fluid on the wall is increasing because it's being condensed or forced into a smaller chamber. That's a very short little period of time, but it's an important period of time. Okay? Now, when we start to see the pressure of the fluid in the chamber is greater than the fluid on the other side of the semilunar valve. So on the right side, that means it's the pulmonary trunk. The left side, it's that ascending aortic area. We are going to see that is when the second phase of ventricle systole starts. And that phase is going to be when ejection of your blood is happening. And it's hard to spell with this. But phase 2 of ventricle systole is when we now see the semilunar valves open, and as long as the pressure of the fluid in the ventricle is greater than the pressure of the fluid in the aorta, we are going to see blood is able to leave. Okay? And then at some point in time, when the amount of fluid still in the ventricle, even in the chamber that's beginning to start to slowly get bigger, as cells begin in the apex area to maybe start repolarizing and relaxing, we are going to see that there will be a time point where the ventricle stops having enough pressure to push blood out and the semilunar valve will close. When the semilunar valve closes, the ventricle is no longer able to push blood out. The ventricle is maybe got a few cells still contracting, but for the most part, most of the ventricle cells are trying to repolarize and relax, and we see a time point of ventricular diastole, time point A, where we're in a isovolumetric relaxation. Okay? And then when the ventricle is no longer got any depolarization, no longer has any cells contracting, that will be when the papillary muscles are no longer even contracting. And if the papillary muscles no longer contract, they're no longer pulling cords on the AV node flaps to stay closed, then the AV valve will open and we will be in the second stage of ventricular diastole, which is when the AV valve is open and filling of blood, venous return, preload is happening. Okay. Now, I will ask you to walk me through a cardiac cycle. All right. If drawing a picture helps you like this, figure out a cardiac cycle, draw a picture. Doop. The other way to do a cardiac cycle might be, and this is my version of what is a cardiac cycle written in the 4 key events. So a cardiac cycle starts over here. Atrial systole is happening. Atrial systole has these sub-events happening. The atrial cells are contracting. The right and left AV valves are still open. The atria ejects blood into the ventricles to fully fill the ventricles. The atrial systole starts to end. The AV valves will start to close. The ventricles are then going to contain the largest amount of blood inside them, and that is going to be known as our end of ventricular diastolic period volume. Again, that's a lot of words, so we shorten it to our end diastolic volume of the ventricle, which is also abbreviated as the EDV. Okay, so this is the first thing that happens when we start a cardiac cycle. The atria starts to, again, contract first. It pushes blood into the ventricles. When the atria is done contracting, the fluid that is in the ventricles, as the ventricles are going to start to see depolarization, and as they start to depolarize, the papillary muscles are going to pull on the chordinae tendineae and close the AV valve. That fluid stuck inside the ventricles when the AV valve is closed is known as the end diastolic volume. It represents the largest amount of blood in the heart, so it might be 100 milliliters. All right, and again, if this is your reference, this is 500 milliliters, so 100 milliliters is probably like a fifth of this, the amount down here. I don't know if I have my water bottle. This water bottle is 300 milliliters, so again, about what I have left inside of it, about the bottom third, that's probably a little bit more than 100 milliliters, but that's about the amount of blood we would want, or a little bit less of this, we would want in the ventricles, the right and the left ventricles, at the end of their relaxation phase and at the end of the atrius pushing a little extra blood into the ventricles at the end of ventricular diastolic period. Okay? Now, atrias are going to enter their diastole period. Atrias are going to relax, but the AV valve is closed. The atrias, as they relax, are going to, again, think about the auricles. The chamber is going to kind of collapse in size because the contraction or the lack of contraction by the cells are going to just kind of flop down. Okay? The ventricular part of the system now takes place. So remember, the AV bundle branch is now spreading the electrical depolarization down through the Purkinje fibers, the moderator band, through the contractile cells. The AV valve closes. Now we start to see depolarization leads to contraction, and we start to see phase 1 of ventricular systole. All the doors are closed. The ventricle starts to get smaller in size, and the blood trapped in that chamber starts to increase in pressure because it's being collapsed into a smaller space and it's pushing more on the walls. That first phase of the ventricular contraction, ventricular systole, is known as isovolumetric contraction. And it's a few microseconds. But it's a period of time where we see the ventricle gets more and more and more of its contractile cells working in unison, it gets enough of the contractile cells working to make the chamber small enough that the fluid trapped in that chamber, the EDV, the fluid, the volume inside the heart, trapped inside the ventricle chambers, can exert enough force that it can then push the semilunar valve open. So the pulmonary valve and the aortic valve. Now, the second phase of ventricle systole is as long as the chamber keeps getting smaller and the fluid in there stays at a higher pressure than what's on the other side of the semilunar valve, the fluid will start to be able to shoot. All right? When the fluid in the ventricle is no longer able to maintain a higher pressure than the fluid in the aorta or the fluid in the pulmonary trunk, the semilunar valve will close. When that valve closes, you no longer have ejection. Technically, will there be some ventricular cells still contracting? Yes, but the number of ventricle cells contracting are probably beginning to, every millisecond, become fewer and fewer and fewer cells. If the ventricle cells are beginning to, again, not be at 100% contraction, but at like 40% contraction, 30% contraction, the ventricle chamber is going to start to increase in size. The fluid trapped in the ventricle at the end of ejecting out, when the semilunar valve closes, is known as the end systolic volume of the ventricle. It's abbreviated the ESV. That volume is, as the ventricles continue to relax and less and less and less cells are going to be in contracting, they're beginning to repolarize and go back to, again, their stage 4, their resting membrane potential. That volume, which might be 30 milliliters, okay, is going to then expand into a bigger area, and as it expands into a bigger area, it's going to push less on the walls. So the pressure of the fluid exerting force on the wall of the chamber is going to drop. All right? Okay. So the end of the ventricle systole period is the closing of the semilunar valves. Okay? The beginning of the ventricle systole period is the closing of the AV valve. When the fluid that is maximized in the ventricle is there at the start of systole, the fluid at the end of the semilunar valves closing, at the end of the systole is going to be the lowest amount of fluid in the ventricle. The difference between those volumes that was ejected into the aorta or ejected into the pulmonary trunk, that volume, in diastolic volume minus systolic volume, so we get the difference that got to leave, that is known as your stroke volume. And on average, our stroke volume is anywhere between 60 to 70, 72 milliliters. So remember I said you have about 100 milliliters in there at the start of systole. That's your end diastolic volume, your EDV. And then at the end systolic volume, you'll be somewhere maybe around 30 mils, and you eject it out something like 70 mils. Okay? And again, that's physiology fun math. So is it the exact number? No, but it's a number that we can all hopefully visualize pretty easily. Now, when the ventricles begin their diastolic period, remember the semilunar valve is closed. Some of the ventricle cells are still contracting. I need that isovolumetric relaxation. I need my chamber to get big again. I need my ventricle cells that were the last ones to depolarize to continue to cycle and finish their contractile phase, finish their repolarization, finish their recovery into resting state. And that is going to be my isovolumetric relaxation. Once all of my depolarization, all of my plateaus, and all of my contractile cells have gotten to a place in repolarization and return to resting membrane potential that there is no more, again, activity of positive ions coming in and trying to activate the next contractile phase, that is when my AV valve will open and my into isovolumetric relaxation is done, because once the AV valve opens, blood is going to start to, with gravity's help and by physics, the equation of flow of a fluid, flow of an air is the going to happen because there's a pressure gradient of high pressure somewhere versus low pressure, and so fluid wants to move from the high pressure to the low pressure. Okay, and that filling will happen of the ventricles, and most of the filling of the ventricle is just by the simple flow equation of I have a pressure gradient and the pressure of the fluid in the superior vena cava, in the inferior vena cava, is greater than the pressure in the right atria, and the pressure of the fluid in the right atria, which is a smaller chamber, is greater than the pressure in the ventricle, and so fluid just keeps filling into the ventricle because of the pressure gradient. Okay? And then I get to a place where I'm mostly full, and I start my next cardiac cycle when the atria start again by the SA node triggering a threshold and an action potential start to enter their systolic period again. Okay, so I need you to get comfortable with writing a cardiac cycle. A cardiac cycle means you're talking about periods of contractile cells contracting, periods of contractile cells relaxing. So you have to have the words ventricle, atrial diastolic or diastole, ventricle atrial systole. For the ventricles, you need to make note that just because they are contracting or relaxing, it doesn't always mean fluid is moving. So there are periods in the systolic state where you have isovolumetric contraction versus ejection, and periods in the diastolic state where you have isovolumetric relaxation and you have filling. You need to make a note of when the most blood is in the ventricle, when is the highest volume of blood there. That's at the end of the ventricle's diastolic phase. And then when you have the least amount of blood in the ventricle, which is the leftover blood, that again, I can't force out all the blood, so I get left a little bit. That is my end of my systolic period of the ventricle, so my ESV. The amount of blood that leaves is my stroke volume, okay? And it represents the amount of fluid that I'm putting into the aorta, and then that's the fluid that in a few seconds will then spread to the major arteries, and then in a few more milliseconds spreads to the arterioles, and it's slowly being divided up. That 70 mils, maybe 3 mils gets to my brain. Maybe 1 mil goes to my right arm. Maybe 1 mil goes to my left arm. Maybe 1 mil goes to my stomach. Maybe 1 mil goes to my right leg, my left leg, and maybe 20 mils goes to my kidneys. You know, so it's being distributed across the body. And remember, it's not being distributed evenly. And because it's not being distributed evenly, we know that the vessels are going to have different diameters, different radius to their lumens to try to, again, direct some blood more to the kidneys or some blood more to the brain versus less blood to your ovaries or your testicles, to your, I don't know, to your pancreas or something, okay? You're going to try to divert the blood where it's needed most depending upon your metabolic situation. All right. If you're running or you're physically active or walking, your 70 mils, maybe 50% of it's going to go to the muscles of your legs, your back, your belly. And then the other 35 mils, 50% of 70, if I do math in public, is 35 mils. That 35 mils then has to be more or less distributed to the kidneys, to the brain, to the eyes, to the arms, to the stomach, to the bladder, to the ovaries, to the testes, to the pancreas, you know, those other places. Okay, so again, that comes back to your arteries, and your vessels are going to then move the lumen size to help, again, distribute that 70 mils, that stroke volume, across all the capillaries of our body. Okay. All right, so if we start to put everything together, what's happening electrically up here is going to then be captured on the EKG. What happens electrically will then be followed by physical changes to the contraction, the chamber size, the fluid amounts, and the pressure readings in our right and left hearts. Okay? Now, question was, when is the most blood during the ventricle? The most blood in the ventricle is when the ventricle is resting and passively filling, and then the atria finishes our systolic period, and the ventricle is still in diastole and accepts a little bit more blood from the atria. So the most blood in the ventricle that you will see is over here at the end of the diastolic period for the ventricle. Okay? And that point in time is normally when you are going to see the AV valve closes, all right? Because the ventricles are about to start contracting. And if the ventricles are about to start contracting, that means a wave of depolarization, represented up here electrically by my, again, QRS complex, that is all happening and about to make the ventricle contractile cells start to contract. And when they start to contract, I need those papillary muscles to hold tight on my AV valves to make sure that as the ventricle makes itself a smaller incised chamber, the blood won't go backwards. It can only go forwards, and it will only push through the semilunar valves when the fluid pressure inside the ventricle chamber is greater than the pressure of the fluid sitting in the pulmonary trunk or the pulmonary artery. Okay? Now, again, the electrical events happen first. So the P wave is going to represent when the atrias are in depolarization. Sodium's coming in, calcium's coming in, the plateau is happening across the entire atrial cells. All right? So the middle of the P wave is usually representative of a time where the most cells are undergoing rapid depolarization plateaus, and the most cells are having calcium being released from the sarcoplasmic reticulum, and calcium is letting actin and myosin interact. All right? The question just asked, why is this picture only the left ventricle? All right. The right ventricle is the same looking picture. The scale on the right ventricle is going to be from 0 to 8. All right? Because the pressure changes on the right ventricle side are a fraction compared to the changes of pressure happening on the left ventricle side. That's part of the reason why your right ventricle is known as your low pressure pump and your left ventricle is known as your high pressure pump. Okay? And let's think about that. My right ventricle needs to pump blood. It's going to pump the same amount of blood as the left ventricle. So this value of 130, this value of 50, that's the same amount of fluid going in the right ventricle, the same amount of fluid leaving the right ventricle. That's stroke volume. But that stroke volume has to go to the right and left lungs. So geography of your body, is that a long way away? Compared to the left ventricle has to pump blood to the tips of my fingers, to the tips of my toes, and the top of my head. Can you see that there's a longer amount of distance that the left ventricle has to push blood to than the right ventricle? Okay? All right. The second part, the left ventricle is going to be pushing blood, again, into these vessels that are all different sizes to try to distribute blood to a lot of different locations, but they want to distribute, again, 1 mil here, a half a mil here, 2 mils here. The left and the right lungs want, again, the blood to be distributed pretty much wherever there's air and blood getting close to each other. So the amount of arterial resistance from vessels in the lungs is way less than what it is on the left side. The other thing of gravity effects, if we're standing up, there's not as much gravity to overcome for the right side of the heart versus the left side. I have to push blood against way more gravity to be able to get it to the eyeballs, to the top of my brain. So the amount of resistance that I have to overcome for the right heart, and again, resistance means if I'm going to roll a marble up a ramp, the amount of height that I have to go, the amount of distance the ramp is to get the marble to the other end, that ramp on the right side is like this big and like maybe a fraction of an incline. The ramp on the left side is going to be like this big, and the ramp might have an uphill of a very steep angle. So when I push the marble on the right side, I just have to flick it a little bit. On the left side, I might have to really shove it, okay? And that force is basically represented by the pressure we have to have. Okay, so think of the pressure as the push that I have to give the marble, and the marble being the stroke volume, to be able to make sure it gets to the top of the ramp, whether that's all the capillaries of the body represented by the top of the ramp. On the right side, it's not as big a deal, okay? And so the right side, if I was to draw this, it would look the same, but the difference would be the top up here would be like 8 millimeters of mercury. And remember, mercury in a bowl, air is going to push on it. All right, let me get a little different color. So as air pushes on mercury, that force makes it rise to a certain height. And if I measure that height in millimeters, that's what I say the force is equal to. The push on mercury to make it rise up to a certain height, that's what force is, all right? And so the right heart has to push on the bowl of mercury to make it rise to a height of 8 millimeters. The left ventricle has to push on the bowl of mercury, and it has to rise up to a height of 120 millimeters. So you see how much of the difference between the left and the right side. So if I was to show you the right ventricle in this picture, it would look the same. The numbers on the graph, specifically on the left graph, would be different. They would be 0 to 8 on the y-axis, okay? And the other big thing we do with the left ventricle, in the heart, we talk about everything from getting blood back to the right atria, and then we just assume, okay, it's fine going to the right ventricle, it's fine going to the left atria, it's fine going to the lungs, and then it gets to the left ventricle. So when you see or read things, it's all about how do I get blood to the right atria, and then that blood in the right atria magically is dropped into the left ventricle. And we kind of talk about those two chambers, and we ignore the left atria and the right ventricle, unless you start to have pulmonary issues, which is problems with the blood flow and the issues of the lungs, creating, again, the right ventricle, then having to remodel. Okay, so now I've lost my train of thought over what I'm supposed to be talking about. Okay, so the whole point of this is electrically what has happened. Movement of sodium ions, movement of calcium ions, movement of potassium ions is going to make contractile cells physically change, contract, go into periods of systole, go into periods of diastole. And as that happens, we are going to capture that by what is happening with the pressure reading, if we put a little probe inside the left ventricle, and if we measure the fluid inside the left ventricle. Okay, so in the middle of the P wave, we know that the atrias are going to enter systole first. The atria pressure is this blue line. And as you see, the blue line is slightly above the red line. And again, how do I make fluid move? I make fluid move because I have a high pressure and I have a low pressure, and fluid wants to move from the high pressure to the low pressure. So because the blue line is slightly on top of the red line, what does that tell you is happening with fluid? It's moving from the right atria chamber into the left atria chamber. And if you go down here, you see that fluid is moving from the atria into the left ventricle. And as it does so, it is going to keep adding fluid into the ventricle, and the amount of fluid in the ventricle is going to reach a higher amount. All right, now the atrias are going to finish their systole. They are going to start to repolarize and rest and go into their diastolic period. And again, we don't see repolarization on the EKG because it's masked and hidden by the ventricles starting to depolarize. And there's so many more ventricular cells that they hide anything electrically the atrias might be doing. Okay, now initially when the ventricles are all going through depolarization and electrically getting excited and calcium's being released, the AV valves are going to close, all right? And so between, again, I need my little pencil, between this time point at 100 and again this time point here, we see a rapid rise in pressure, but we see on this line here that there's a flat line for there's no fluid movement. Right? So the takeaway is this first phase of ventricular systolic period is there's no movement of fluid in or out of the ventricle. So it's an isovolumetric phase. But what's happening with the pressure is the red line is shooting up, and it's shooting up because the chamber is getting smaller, and this 130 milliliters of blood, this end diastolic volume in this chamber, has nowhere to go, and it's going to be compressed. And as it gets compressed into a smaller chamber, it's going to start to elbow and push on the walls with more force. That is known as again, this little point in time up here on the ventricle systole, that is going to be that little point in time known as ventricle systole isovolumetric contraction. Now, the ventricles are going to eventually have the blood inside that chamber reach a pressure that is going to then be more than the pressure of the fluid in the aorta. And when that happens, our aortic valve opens, and we will start to see the second part of ventricular systole, where we are ejecting, and again, it's hard to write, ejecting out fluid, all right? And as we eject out the fluid, the amount of fluid in the ventricle starts to drop. Now, one thing to see here on this graph is the pressure continues to go up for a little bit. Remember, the pressure still is going up despite fluid leaving, because chances are we are still seeing, like, this is probably maybe where 80% of the ventricle cells, I'm just making this number up, but let's say that's where 80% of our ventricular cells are starting to contract. This is maybe where 90% of the ventricular cells are in a state of contraction. Well, this is where all ventricle cells in and around the right and left ventricle are actually contracting. And this is where they are probably making the ventricle chamber the smallest chamber possible. And the fluid in the chamber is, again, being compressed and able to leave, but it's being compressed into the smallest amount of space. Now, as the hump happens, what's going to start to happen over here? Some of my ventricular cells are going to start to hit their repolarization, hit their resting membrane potential, and so some of my cells are going to start to relax. So maybe 95, if I have 100 ventricle cells, only 95 now are contracting, and now only 70 are contracting. And the amount of fluid in the chamber has decreased so that between the number of cells no longer in a state of contraction and the chamber starting to get bigger, the less fluid in there is not as forceful, and so the pressure drops. And when it drops to a point that the blood now in the aorta is now greater in pressure on the semilunar valve than the fluid in the ventricle, my semilunar valve closes. Okay? And at that point, my ventricle is known to be entering its ventricular diastolic period. Has every single cell finished relaxing in the ventricle? No. So the first part of ventricular relaxation, again, is a period here of no change in fluid, because the AV valves are still closed because some ventricular cells are still depolarized, still repolarizing, still electrically active, and I need to keep my AV valve doors closed. All right? And I still continue to see, again, if this was like 75 right here, 75 out of 100 ventricle cells are contracting, well, here 50, here 30. And it isn't until I get to 100 or 0 cells contracting, 100% of relaxation. And again, that's at the end of the T wave. So all of my ventricles have repolarized. All of my ventricles are now in phase 4. All of my ventricles are in, I am not touching actin and myosin. Troponin is, let tropomyosin come back on top. Is stuck in the sarcoplasmic reticulum or being pushed out of the cell. So there's no calcium binding to troponin. That is when the second part of ventricular diastolic starts, and my AV valves no longer need to be stayed closed because they can open and start to let fluid come back in and fill the ventricle chamber. Okay. So what I like about this picture is it brings in what is happening electrically and what is happening in a cardiac cycle, and it puts more numerical value to the cardiac cycle is going to be a change in pressure of what is the blood in the chambers doing, pushing on the walls. What is the change in actual blood in the left ventricle chamber, and seeing that the blood is coming in when the ventricles are relaxed. The blood is leaving when the ventricles are contracted. And there are these little time points at the beginning of contraction and the beginning of relaxation where until the pressure is enough or the pressure is not there anymore, I have isovolumetric contraction and isovolumetric relaxation. All right. Now, it's going to take you a little bit of time to let all this marinate, to let all this come together. So what I'm going to suggest you do is work through these 3 slides, all right? Whether it's this picture, whether it's these bullet point words, or whether it's this picture. You need to come up with a way that you can it would be write and draw on a paper test, write and draw out what is a cardiac cycle, what are the phases of the cardiac cycle, and what are these important kind of midpoint and items within the cardiac cycle that we're looking for. So we're looking for isovolumetric contraction, isovolumetric relaxation. We're looking for end diastolic volume. We're looking for when we have end systolic volume. And I need you to be able to put that together into a essay, a short essay, a short answer for a question asking you describe with correct terminology and points in time or points in phases the cardiac cycle. Okay. And then when we're ready, we'll do this Friday. Once we then understand what is happening with one heartbeat, then we want to start to look at, okay, how is the system doing over a few heartbeats? Because remember, if I try to capture how's your heart doing, and I only take a sample of one, what if you hiccuped? Or what if that was the one heartbeat that something screwed up and it looks terrible? Remember, if I want to see how your heart's doing, I want to get a sample of heartbeats, and I want to see a sample of cardiac cycles, and I want to see a sample of stroke volumes, a sample of the system. And so when I start to look at a sampling of the system, now I can start to look at your heart. Actually, maybe it has about every 100th beat a screwy one because you hiccup, you cough or something, or you fart, all right, and you screw it up or you hold your breath. But for the most part, the heart over a sample of 100 beats or over a minute, this is how it does, this is how it's working, and it does great or it doesn't do great, okay? So that's where I want to leave you right now. One more question to look at. Is the stroke volume about 70? It can be a little less, it can be a little more. Again, we were making the math easy for us, and I said 100 minus 30 was 70. In the picture above here, the stroke volume is 130 minus 50. So what's 130 minus 50? Again, I don't love public math, but that should be 80. And again, in some cases, it might be 120 minus 50, so then it would be 70, or 110 minus 50, it would be 60. So if you do your math real quick, I'm sitting here at rest. My heartbeat is 90 beats a minute. And again, I need to get my calculator to do this. So if I have 5 liters of blood, which is 5,000 milliliters, okay? And my 5,000 milliliters of blood comes from 90 beats, then actually my stroke volume per my calculator on my phone is 55 milliliters per beat. So my heart is pushing out 55 milliliters, and it does that 90 times in a minute to give me 5 liters of blood right now to get to the capillaries in my brain, the capillaries in my fingers, the capillaries in my toes, the capillaries of every single capillary bed in my body. Okay? And that's the right heart has to send 5 liters of blood to the lungs, so that way the blood coming back is then 5 liters for the next minute to leave the left ventricle. Okay? All right, when you say describe, could that be a drawing? If you are taking a paper test, yes, you could potentially do a drawing as long as you put in the drawing the key terms that make me see you understand that a cardiac cycle means you're talking about when is an atria and systole, an atria and diastole, a ventricle and systole, a ventricle and diastole. When do I have ventricular isovolumetric contraction, ventricular isovolumetric relaxation? When do you have EDV and ESV? So if you go back to this slide, I think the bolded terms are the terms I want to make sure if you do it by picture, you make sure those terms show up in your picture. Okay, so the bolded terms here, if you're going to take the paper test in person and you're going to plan to do this test and answer this question with a picture, make sure these terms are represented with arrows to this is when this is happening, this is when this is happening. And again, for some of us, this picture from the book is going to be the one that makes most sense to us. For some of us, I don't want a picture. I just want to draw it out. I just want to write it out. And then for others, this is going to be the most sense. Again, you don't have to put, you don't have to put this part up here. You just need to do maybe some of this part or do this part and then kind of put in this is when this is happening. This is when this is happening. This is when EDV is. This is when ESV is. Okay, you can make it work. Normally, I'd say visit the tutor or visit the PAL and look over your work and maybe make sure that you have it in a logical flow. But since the tutor, again, didn't take my class, although Meg has seen enough of my students, she might be able to help you. You might be able to get her to help with this. Otherwise, work together and share each other's pictures, share each other's writings, and then try to see if you could grade someone. Do they have all the things that I'm trying to remember to put in my essay or my short answer? Okay. I'm going